but it's kind of an essential glioblastoma transcript, and if you knock it down, it hurts the cells. And what they showed is that in both those cases, if you target these with Cas13, you actually get cleavage of ribosomal RNA, and in the case of the EGFR-V3, you can actually see death and all these other phenotypes. I think it's not perfectly well controlled. There's actually a small debate on Twitter. You can see that I wouldn't say it's a debate, we comment on. So the take-home question is, it does happen in mammalian cells to an extent. It's context dependent. We still don't know exactly why it does not happen in these other contexts, but the mammalian cytosol and nucleus is very different than the bacterial cytosol. And there's all these different factors. There's RNA helicases ripping things off. There's translated ribosomes. There's just stuff that binds the RNA, and it's dense. I mean, all cytoplasms, or metabolic things are dense. So, yeah, it's cool. You can talk about maybe the application.
Yeah, I mean, I think the collateral is super interesting. We originally concluded that, it must not be happening in all mammalian cells because of this, one experiment on F293s. But I think we started getting reports in after that paper that, some people saw Cas13 was toxic in certain cell lines, or, some people could actually, visually see, RNA cleaved on gels in certain cell lines that they were testing. And so I think we've come to the conclusion that, even human cell lines can vary in terms of how, their cells are organized and how the buffers in the cell are. And so it's kind of cool that you can start to see collateral used as this way to blow up cancer cells, essentially. Is what this paper was showing. It's kind of a neat therapeutic angle for it.
Yeah.
Yeah.
And we actually have a question or two about that in a few minutes. So
yeah.
We'll wait on that. So why don't we go to the next question?
Hi, my name is Anna. We were wondering why gRNA mismatches increase the specificity of Cas13a.
Can you just repeat it's like the miking? Why did gRNA? One last. Sorry.
Why the gRNA mismatches increase specificity of Cas13a?
Why the gRNA mismatch? Specificity. The synthetic mismatch you're referring to specifically.
Yeah, so what we realized in the original Cas13 study is Cas13 was very specific to its target, but it could tolerate a single mismatch. So, double mismatches or triple mismatches or more would completely kill recognition of the target and cleavage activation of the hepN domains. And so when we were thinking about, how could we use this for genotyping, if you're trying to detect a specific SNP, your target and the target with a SNP are going to be only one mismatch apart, and so you won't be able to distinguish them. But we realized that we introduced an additional mismatch in the guide RNA, right? The guide RNA will only be one mismatch away from the SNP you're trying to detect, but it'll be two mismatches away from the wild type sequence because you'll have the SNP and the additional synthetic mismatch. And so that allows you to actually get distinction between a target with a mutation and a target without the mutation. And so it's just this clever trick to take advantage of Cas13's way of recognizing, targets with mismatches to allow for actual human genotyping and other cool applications. Yeah.
And there were some papers on using kind of similar approaches with Cas9 before that, not for genotyping, but actually that Jim Collins on. So we drew from that a little bit too.
Okay. All right. Chang,
hello, my name is Chang, and I was wondering, why do different Cas proteins have different dinucleotide specificities?
Why do different Cas proteins have different dinucleotide specificities? Yeah. I assume you're referring to the different Cas13 orthologs. We don't totally know. I mean, it's kind of interesting to think about why these, if you think about the pocket of the hepN, actually endoribonuclease site, why it would accept a uridine versus an adenine versus two adenines. But if you think about it, there's going to be some preference for when it's actually docking in the nucleotides. The problem here is that I can, kind of postulate, but no one has really crystallized or solved the structure of a Cas13 in complex with a target actually in the hepN site. There are good tertiary complexes where you have the Cas13, the guide, and the target. Those are bound at basically the CRISPR RNA target homoduplex. But no one really has a good in-action picture of it cleaving. And this is something that we're really hoping that someone does it. We're not actually actively working on it, because understanding that mechanism could lead to more colors of Sherlock if you think that we can rationally change these things. So it's definitely a structural aspect, and it's something probably around the HepN site. But what specific residues are mediating that interaction? I wish we knew.
All right, great. Jeff, do you want to ask the next question?
Yeah, so my name's Jeff, and so regarding the Cas13a off-target effects that y'all looked at in the mammalian cells, y'all reported 0 off-target effects in the cell lines you used, and I was just wondering, like, that seems unreasonably low. And so I was wondering if y'all looked into that anymore and like found that there actually are, or is it actually like 0 off-target effects in those cell lines?
I guess this is referring to like RNA knockdown in cells. Yeah. I mean, when we initially did these experiments, I think we were also pretty surprised about how specific it was. I think if you look at the distribution, like with RNAi, you're used to seeing kind of a cloud of transcripts because they're randomly perturbed between targeting and non-targeting guides. But when you looked at Cas13, you really saw this much thinner, tighter distribution where the transcripts really were not being perturbed by Cas13 sort of being in the cells. And so I think, part of that is it's sort of an orthogonal system. So when you're trying to use RNAi and use the dicer and those pathways, they're endogenous and they're in some ways purposely leaky, so they can sort of regulate endogenous RNA regulation in the cell. Versus Cas13, which has been sort of evolved to be rather specific towards sort of viruses and sort of the warfare that happens in bacteria versus viruses. And so I think our understanding is like when you port over these orthogonal systems that don't normally exist like in mammalian cells, you can get fairly good specificity. But I think we were also surprised just because we had assumed just RNA interactions are a little bit more permissive than DNA binding, just because there's also non-canonical base pairs that can form and whatnot. So I think we're starting to see that it can be cell line dependent as well. And so I think seeing the specificity in other types of eukaryotic cells would be very helpful to have a better understanding of that.
Yeah,
So,
I think two other small points, I guess, as a moment. I mean, off targets, these are significant off targets. Transcriptional states between cells and different transfections are going to be different. And so, we're taking 3 different bioreps and looking for anything that's significantly different. So, nothing changing is kind of a subjective measure. The other thing that I think is important to note is that we're always comparing targeting and non-targeting guides. And that's what we did in our 2 papers. I'm pretty sure that's what Patrick did in his paper. Mm-hmm. Those are the papers that they specificity, right? Mm-hmm. And, when you put these tools in, you're going to change a lot, right? Because you're expressing a new protein that's strongly expressed. So it's not like, this is not doing anything to the cell. When you put Cas9 into a cell under a strong promoter, you're going to change how things work just because you're putting a new plasmid in and you're transfecting. So I think that's always also important to keep in mind. All of these molecular tools have caveats. I don't think they're big or even like notable. That's why we don't really talk about them. But you're not, the cell is not exactly the same as another hex cell in the dish over, next door that hasn't had anything done to it.
Right. I think some of these questions stem from the fact that I was saying that 0 effects is just a shocking thing to report, right? Usually there's some small off-target effects, and so we were just having a discussion about that in class, and I think that's where some of these questions are stemming from.
Yeah, I think 13A in the RNA editing.
Professor review paper. There was I think like 5 to 10.
Yeah, I think a small amount.
Right, in editing paper. Once you got the second version, it seemed to work much better, obviously, for the repair.
Yeah. Oh no, we're talking the first figure of the Cas13b paper.
Oh.
We compared it to Cas13a and the RNA-seq knockdown analysis.
Yes.
There was a couple off targets with 13a.
Okay.
Yeah.
All right, Megan.
I'm Megan, and I was wondering if you think it's possible to directly detect RNA cleavage with Cas13a rather than using the collateral cleavage in the reporter systems?
So just to make sure, I guess we understand correctly, so directly detect sort of target RNA cleavage rather than collateral activity?
Yes.
Yeah, I think that is a little difficult because it's actually this, right? It's the same domain that does the on-target cleavage as it's doing the collateral cleavage. And in fact, the on-target cleavage in a way is the collateral cleavage. Because if you imagine the RNA target gets bound by Cas13, and then the Hep domain, which is like outward facing, is kind of like these scissors that just get activated, and it'll cleave the target molecule that's bound. It'll also cleave target molecules that are unbound, and it'll also cleave anything else in solution. So if you wanted, one way to maybe do that is to have some sort of labeling assay where maybe you could, have the target that gets bound to the Cas13 somehow get covalently stuck, and then maybe like a photolabile thing to see like direct cleavage, and maybe you'd have to kinetically control it, so it's like you have a very short reaction where it's like induced and then stopped. But in general, the collateral cleavage is the same as the target cleavage, so I think that is hard to separate. Of course, we've done experiments where we label the on-target RNA molecule, so you can look at fluorescent, like just look at that molecule getting cleaved in solution, but it's going to be a mixture of the on-target and the collateral cleavage still.
Yeah. You do a single molecule study, maybe. You could kind of do like FRET also.
Yeah. So like if the RNA, maybe you have like the protein has 144 and then like a FRET pair on the RNA molecule that comes into contact into it, and you can see like fluorescence when like they associate, and then you can see that maybe dissociate when it gets cleaved. It'd be hard to separate that from like unbinding also.
Yeah. I mean, the other thing you could do is this actually depends on magnesium binding, but you could do like a turf setup where you get like a bunch of Cas13s on a slide, stick them there, let them bind to like a doubly labeled, like a 5' and 3' different color labeled RNA, and quench in some magnesium and see it cut. And then you could also have a third color collateral for it, and you could see that going through. Yeah,
Actually,
a lot of...
the third option is there's this new technology, I guess it's a few years old, but it's like ultra high speed AFM.
Oh, yeah.
So AFM is like atomic force microscopy, and it's basically, this technique that has a very ultra fine, like almost atomic scale tip, and it can scan across like a 2D surface area, and you get a picture of the 2D surface when you're scanning. And people have shown that you can actually image proteins this way that are deposited on the surface by scanning across the proteins. You can see the shape of the proteins. But what happens if you do it really fast, like hundreds of thousands of times, I think a second, you can start to actually make a movie of what's happening on the surface. And people have shown you can see molecules. And so like a year ago, I think, was it Osamu Nareki, which is a structural group in Japan, they published a video of Cas9. You literally see Cas9 moving around, and it associates with a DNA molecule, and then cuts it, and you see the DNA dissociate. And so that could be another way, actually, just to literally see Cas13 cleaving its on target versus like other RNAs that are not associated.
Yeah.
That could be one way. Cool. Yeah.
Very, very cool. Great question.
Hi guys, I'm Victoria Valensky. I'm the one who is bombarding you with emails, so I appreciate you answering. So my question is, how did you know that changing primer concentrations would make Sherlock quantitative?
That's a good question. We didn't. I mean, I think that when we thought about the whole Sherlock workflow, I mean, Cas13 is generally, if you just do a titration of RNA against a Cas13, you can see the collateral is fairly quantitative, but RPA, it's like an asynchronous exponential process. It also has a lot of spatial variability. It's kind of a weird reaction. And so we thought maybe we can kind of tune this a bit. And the primer concentration is the easiest aspect to just go along and see, if we obviously have too little, it's not going to run, but if we have a limiting amount, maybe we'll have some sort of, quantitative effect. And so that's what we found, that when we kind of just get in that sweet spot, it makes it much more quantitative. RPA itself, though, is quite an annoying reaction. For other reasons, we're actually starting to somewhat switch away to a different isothermal application called LAMP. But yeah, I think we didn't know, but we did the experiment.
Nice.
I mean, the main issue was the preamplification was saturating, right?
Yeah.
So like when you preamplify, you take away the quantitative nature because everything just becomes really highly amplified. So we were brainstorming ways to limit the preamplification.
Yeah.
So that was one of many ways, I guess, we tried to limit the RPA.
Great. Gwen.
Hi, my name is Gwen, and I was wondering if you guys have used Sherlock to detect more clinical pathogens, like in a hospital setting, such as like MRSA or staph. which could be used to maybe prevent infection.
So just to make sure that we got that clinical setting Sherlock for, did you say staff or specifically?
Staff or MRSA, just because like a lot of times like patients get infected from like dirty environments. So do you think it would be possible to use Sherlock to do that?
Yeah, I mean, certainly be possible.
Yeah, I mean, we've done clinical sort of, carbapenem resistant, gut bacteria. We've shown detection for like tick-borne diseases, other viruses. We've done Zika, dengue. We haven't done like nosocomial infections, like things you pick up in the hospital yet, but there's no reason, you shouldn't be able to design an assay for it. We're actually working on a new paper that's going to release a guide design tool, so you can easily put in like a new organism's like genome, and it'll design the best guides using like a machine learning model that's built on like thousands of guides to sort of make it easier to just deploy these tests. And in there, we also did a clinical application for a specific type of blood cancer. I think that would be like a really good test. There's also like this new like fungal infection, Candida auris, that's been spreading, and so there's been brainstorming about like, can you detect that like happening in the hospital? Oh, what's up?
No, I had added that to her question. She just didn't ask that part, but I was specifically wondering about that particular infection.
Oh, yeah. So we're brainstorming both ways that like you can use Sherlock to detect it, but also ways to use Cas12 or Cas13 to even combat it, right? Because it's so hard to get rid of it. But if you can imagine targeting like a nuclease that can just shred up its genome, that would be great. I think delivery, of course, is the main issue. Like, how do you get these, nucleases into the cells? But,
yeah, I think, there's tons of different applications of Sherlock, both in ID and more generally. I mean, we definitely can't do them all, but actually it's 2 weeks ago maybe, we released a protocol actually for Sherlock. It's online. It should be open access, although Nature Protocols may not have gotten that fixed. But you can actually go online and see how to do Sherlock yourself. The biggest barrier there is always basically getting the Cas13 protein. It's not commercially available yet. If you have access to an FPLC, you can make it pretty easily. But we're hoping that there'll be some good commercial providers to get it kind of inexpensively. But yeah, all of these different bugs, these different pathologies, ag. We even have a collaborator who's using it for environmental DNA. You can like take like a trout or something. It smells actually. It smells. And you can swab like the mucus from it with a Q-tip, and then you can detect, like, figure out what type of fish it is. And you can even do that from like reservoirs, like just they're shedding nucleic acids. So yeah, there's a lot of cool stuff you can do with it.
It's very cool. Angela.
Hi, I'm Angela. You guys were able to create clinically or catalytically inactive Cas13A to do live cell imaging of transcripts, and I was wondering what sort of application that could have.
All right. Can you repeat that one more time? The connection's a little bit spotty.
You guys were able to create catalytically inactive Cas13A for live cell imaging of transcripts, and I was wondering what the application, like clinical or non-clinical, that would have.
Yeah, I think the main application for that is just to really study RNA biology. RNA is remarkable because it's sort of involved in every step of like sort of the cellular pathway, from transcription to nuclear regulation to cytoplasmic and mitochondrial regulation. And so being able to visualize like where RNA goes, how it's shuttled from different parts of the cell, how it's shuttled from cell to cell. There's all these new papers showing that neurons can communicate through RNA using vesicles for like communication. So I think there's been a lot of interest in sort of just better RNA imaging tools, because the current ones rely on having to endogenously tag the RNA with like, something that you can recognize, like MS2 hairpins, so then you can come and image based on the hairpins. And so having something where you don't actually have to perturb the cell to engineer it so you can image it is really powerful, and that's why we wanted to show Cas13 for that purpose. With that said, it is somewhat hard to get the sensitivity you need. So when people actually engineer their cells to add like MS2 tags, they actually add many MS2 tags, so you can get many copies of a fluorescent fluorophore per molecule. With Cas13, we're usually targeting just one guide to the transcript. One thing we haven't really played around with is if you had like 20 guides, you highlight the transcript of Cas13 to try to image the RNA. Would that work better? But yeah, there still are some issues with sensitivities. I think people are trying to improve it. There is literally a science paper, I think, 2 weeks ago that was trying to use Cas13 to image RNA that is being transcribed from the genome. And so that's like another cool application is can you image like RNA and DNA in co-association as transcription is happening. And they were doing that for their biological problem.
Yeah, ironically they were targeting an MS2 repeat.
Yeah.
But yeah.
I think they probably had like that the cell line or
Yeah,
but,
I mean,
yeah.
it's a reviewer experiment probably.
Yeah.
Another note about the catalytically inactivated Cas13 is that, it's also a cool idea that what if you could not only like sense a transcript by binding to it and, having fluorescence, but also actuate on that. So if you have a specific transcript, that's being expressed in the cell, could you reconstitute some sort of transcription factor or protease or caspase that could then do something inside of the cell, like turn on a different circuit or kill the cell or, there's a lot of different directions you can take that. And I think that's one of the exciting things about Cas13 that we really were thinking about for a while. Now that we can sense RNA in vivo, what could you do with that? But as Omar mentioned, there are a lot of technical, hurdles to get there.
Absolutely, no, but it's very cool. Faye, you have a question.
Hi, I'm Faye. I was wondering if you have any specific ideas for using Sherlock for ecological or environmental applications.
Yeah, I mean, we don't have specific projects going on for ourselves, but we actually do have collaborations for like these environmental and eDNA. So one thing you can do is for ecological applications, instead of having to count all of the fish in a specific reservoir, you can use a proxy measurement like the amount of a specific type of DNA, and that's much easier. You can sample that much more rapidly, especially for rare species. Now, these are species that you have to have a good specific nucleic acid sequence that wouldn't have crosstalk. You don't need a whole genome, but you need some sort of marker there. But you can imagine if you find a new species and you sequence part of it, and then you could make Sherlock probes and look for that species in other places. So this kind of ecological tracking aspect is very valuable. And of course, you can also use it to find pests and pathogens in agricultural applications. So if you have an outbreak of an agricultural pathogen, you can then use that for surveillance. So any of these applications where you need to be able to track, nucleic acid signatures, which is basically anything that's alive, or viruses too, you can think about these different ways to use it. And the nice thing is you can do it in the field with a lateral flow strip test. So that's a really nice way to kind of do it.
Yeah, so we actually have 3 questions now about the lateral flow strips. So, Lucas.
Hi, my name is Lucas. I had a question about the lateral flow strips, about why they're not being adopted clinically and kind of what those hurdles are to get it in a clinic or a clinical setting.
Yeah, so there are efforts to try to bring it to the clinic. We ourselves have a collaboration with for the cancer sort of blood testing. The challenge there is for a certain type of leukemia. It's called acute promyelocytic leukemia, APML. Is the patients are very sick. It's almost like they have a bacteria growing in their blood, but it's cancer cells. And if you don't do rapid detection in a matter of days, when they present, they actually can die. And so like doing like next generation sequencing or the cancer tests that you put out and send out to the hospital and take about a week to come back can actually not be ideal. And so there's a need for a simpler test that can be done at the hospital, not necessarily like tertiary hospitals, but even local hospitals that don't have qPCR machines or more complex testing labs. And so the ideal case there would be a lateral flow test that could literally test for the most common sort of fusion transcript that occurs in APML. And so that's sort of one collaboration where we've actually developed a test, and they're trying to take it forward to do sort of a prospective study in the hospital on patients as they come in. And then in terms of like sort of a commercial product that's also in the works, there's a company, Sherlock Biosciences, that spun out from MIT and the Broad Institute to try to make products that can actually be used by clinicians. And there, they are trying to commercialize the lateral flow strip, and the challenge there is basically how do you build sort of a, I guess, product, like a physical product that can house the lateral flow strip, where you can actually add sample, and then when this reaction is incubated, have it transfer to the lateral flow strip. And so that might seem simple in reality, but making that work by anyone and be foolproof is actually a challenge because in the lab, we set up PCR tubes and we let them incubate on a thermocycler, and then we pipette it to a lateral flow strip, and actually making like an easy to use, essentially user friendly product is kind of a challenge there. So that's what's being worked on. Yeah.
So I had a question about the lateral flow strips themselves. It seems like this isn't something that you can just buy off the shelf and use, that you have to prepare the strip for Sherlock, is that correct?
No, so the strips that we actually use in the initial paper are something you can buy. Well, I mean, you can't just pick up at CVS, but they're from a company called Millennia. So you can actually just purchase them. They're not, so if you read into the paper a little bit, we're actually not using the strips like they're really intended to be used. We kind of reverse engineered them in the sense that they're supposed to detect an antigen of the first line, and then that recruits, basically you have a line of streptavidin, and then you have anti-FAM antibodies that recruit gold. And so that would be your first line, your action detection line, and the second line of the strip would be, oh, the strip works. We totally flip that. We grab all of those antibodies at the first line with our RNA reporter, and then if the RNA reporter is cut in the previous reaction, those antibodies can make it through to the second line, which is now our result line. But those strips are actually available. So because of the kind of reverse nature of them, we've been working on making our own lateral flow strips, and there are different companies that can actually do this commercially for you, Mologic, DCN. They're still not that difficult to make. One of the things that we really like to use that for is that you can imagine multiplexing on strips. So if you have multiple lines, you can actually combine the multiplexing capability of the Cas13s that we demonstrate with the lateral flow, and we've shown that we can read up to 3 different colors on the strip, which is kind of cool. That way, we don't do the inversion, we just look for depletion at the strip line. But yeah, I don't think there's a barrier. The real big barrier that Omar talked about is that it takes time to make a product and get it ready and make sure it's totally 100% accurate and precision, everything.
Sure. The only reason I asked is I actually teach a lab class that is devoted to doing CRISPR, so the undergrads actually do it. But after reading about Sherlock, I was wondering if maybe they shouldn't try the Sherlock instead of the Cas9 technique. And I was just curious if we would have to prepare these strips or if we could just purchase them. So it sounds like they're out there.
We can send you the link, yeah.
Well, thank you. Well, so you actually just answered the next question about doing multiple targets on the same strip, so thank you for that. Colin, you have a question.
Hi, I'm Colin. I was wondering which Cas13 system do you think has the most potential for additional uses, like you said before, changing or making it catalytically inactive, and instead of doing that, doing something else, which system do you think would be the most useful?
Oh, I think it broke up through all of that.
Yeah, I'm so sorry. You're gonna probably speak
Try again, louder.
Okay, so which Cas13 system do you think would be the most useful in the future, and which are you most excited for?
So I think I heard most of that. Which Cas13 system would be useful for
in the future for?
Yeah.
Just in the future in general. And which one do you think is most exciting? That's.
So I think, maybe we can both answer this, but I'll give my answer first. I think the RNA editing sort of system of where we take Cas13b and add an adenosine deaminase, or as we recently published, an engineered deaminase that can do cytosine deaminase, can be the most useful therapeutically, which is what I'm excited about, because there's a need for reversible genome editing as well. And so, a lot of, for permanent gene therapy, you think about a lot of rare diseases where someone has, a mutation that causes metabolic disease or, liver disease. But there's also a lot of mutations that you might want to treat almost as a small molecule. Like imagine you take a pill and you get the mutation when you need it, but if anything bad happens, maybe you take the pill away and you don't have a permanent gene edit that could be, long-term bad for you. But also, there are a lot of edits you can do to control pain, for example, to control inflammation, like if you get flares. You can also use it for regeneration. So imagine you get acute liver failure and you want to regenerate hepatocytes. Well, you don't want to make a permanent edit for that because the hepatocytes will proliferate forever. So there are actual RNA edits you can do to repopulate a liver or a heart after a heart attack. And so I think the really downstream things that you can start doing in RNA editing, I think is really powerful. And it also starts to lead to the future of there are all these protective variants you might want to install in someone, for example. So people talk about ApoE4 to ApoE2. So if you have ApoE4, you have a 10x risk for Alzheimer's, whereas ApoE2, you have a much lower risk of developing Alzheimer's. You might not want to make a permanent ApoE2 mutation. Scientists will say they have no idea what the long-term effects would be of that. But if you could have ways to do temporary mutations where you can take it, and if you ever find out that, oh, this is actually a bad idea, you could stop it, that would be really useful. And there are a lot of other variants stronger bones, better better hearts, where you get better longevity and heart function, neural cognition, basically for every organ you can find edits that promote health span and longevity. And so I think having tools where we can start to play around with that is actually really cool too.
That's probably one of the cool ones. I think another kind of totally different angle of castor teams is using them to understand basic biology. So if you guys are following the needle on RNA biology, there's this whole field of epitranscriptomics, which is basically just like the epigenome, RNA is modified in all these different ways. And it's kind of the Wild West because it's hard to detect mutation or the edits, these different modifications that happen. But even when we can detect them, we don't know what they're doing necessarily. We don't know how they're interacting with native machinery or what proteins they're recruiting. So I think the ability to take Cas13 and take the different epitranscriptomic machinery, the readers, the writers, the erasers, that basically can modify and read these edits and use them to recruit and understand what all of these different things mean. I think that's going to be a very neat area to kind of unlock this whole new area of biology. Because really there's these labs working on it and they're just building these foundational tools to have something that's equivalent to all of epigenetics. And it'll be cool to see how Cas13 can help out with that.
I like that answer because my lab's actually funded to look at M6A in embryonic stem cells. But don't worry, I'm not going to steal your idea. Marco.
Hi, my name is Marco. Have you tried using Cas13A to modify RNA in multicellular organisms with viral vectors, and what would be challenges to the feasibility of this?
The Cas13 in multicellular organisms, do a mouse, like a Cas13 mouse, maybe, or I remember. Well, if you're dealing with an actual organism model, there's two ways you can do it. One, you can actually make a Cas13 mouse that expresses Cas13 everywhere and then deliver a guide, or we can deliver Cas13 into a regular mouse using RNPs or viral vectors, etc. In that first one, the challenge there is you just have to make mouse, which isn't that hard these days. It's a little expensive. And then you kind of have to think about, well, how do you deliver the guide and where you deliver it to? And what do you also want to do? We're talking about Cas13, you could be knocking something down, you could be editing. So I don't think there's that many challenges associated with that, maybe getting the guide to be specifically where you want and making sure you have long-term knockdown, depending on what you're investigating. But I think the second potential option, which is delivery to a actually edited organism, is deliveries are hard. So Cas13 is big. All these CRISPR proteins are quite big, and so if you want to package them into things like adeno-associated vectors or AAV, it can be very challenging. So one thing is we would need to find smaller Cas13s potentially to be able to package that and actually have that expressed. Another thing is that how long do you want it knocked down? Knockdown is going to be transient, and then if it gets AAV, it's episomal, if it gets diluted out, you're not going to have knockdown anymore. On the other hand, lentivirus, that can integrate, but then are you going to have enough expression? Because these things can be very dependent on the amount of both protein, and as we've actually seen, much more guide. So there's a lot of questions on how can you get it efficient enough. I think people have been using Cas13D for, I've seen a little bit of data on Cas13D in mice, just to knock certain genes down, and it can be, somewhat effective, but it really also depends on your application, because, if you get 10% knockdown, is that going to give you a phenotype, or do you need 99% knockdown, and in which organ? So, yeah, I think there's a lot of challenges in translating and taking these things kind of up the hierarchy of from hex cells to something that's put in a human. And I think a lot of people are starting to kind of approach that.
Sure, but with all the things that you mentioned, Omar, that you're excited about, I mean, that's sort of the next step, right? You need to get to actually doing it in vivo.
Yeah, trying to figure out the best way to get these systems actually to the tissues you want is a challenge.
Yeah.
I know we've been playing around a lot with AAV, but there have been size limitations. So there's questions if we find smaller Cas13s, and Cas13D actually fits pretty well. When you have the 8, I guess it's a little long. So there have been efforts to find smaller versions of these enzymes. And,
Great.
Yeah.
Okay. We're almost finished. I know you have a lot on your plate, so we have a multi-part question here though about Sherlock and cancer.
So our question is between two people as well, Lucas and Gwen. So first part of the question is it possible to use Sherlock in vivo to label cancer cells and target them, basically they're labeled, and then the immune system can target those cells based on that label?
Yeah, so I guess the question is, can you use Cas13 to label cancer cells? Yeah, there could be ways to think about that. So the first step would be definitely how to target the cancer cells. And there would be 2 ways. One is if there's a specific mutation you want, you can definitely design Cas13 to sense that specific mutation in like a transcript. The other option is if you can identify an RNA marker that's only highly expressed in the cancer cells, you can then go after that transcript. So those would be 2 ways to at least have the targeting side down. And then the second would be, how would you mark the cell for, I guess, destruction by an immune cell? And this would tie back to a little bit about what we were talking about earlier. Can you actuate certain outcomes based on the transcript binding event? And so, one thing you could imagine is you have a split Cas13 system where it reconstitutes on the transcript that's highly expressed in the cancer cells. That could reconstitute some sort of like protein peptide that can then get displayed, and it's like very specific to just that cancer cell, for example. That would be one way to do the labeling. And so that would also be cool. We of course have been thinking about ways to reconstitute a caspase, so then you just actually directly kill the cancer cell. But I think labeling would also be cool. There's definitely a lot of interest in being able to do that. So,
I think he hit on your other questions too. So, Bonnie, why don't you go to your question?
Okay. Hi, I'm Bonnie, and I was wondering if it's possible to create a form of the Cas protein that is inert to the immune response.
You broke up at the very end. Inert to
the immune response.
The immune response.
Yeah. No, this is a great question, because I mean, the immune response is, especially for these things that if you're expressing for a long time, which you might have to do with Cas13, is going to be a big factor. So just to answer directly, who knows? But when you're dealing with the immune response, you have 2 different axes. You're going to have a humoral immunity, that you'll have circulating antibodies, and you'll have the kind of cellular presentation of different epitopes when they're actually in the cell by MHC. And that first one, the antibody response, that doesn't matter as much. I mean, if you think about it, that's only when these things are outside of the cell, really. And if we're packing them inside of a virus, I mean, the protein's not actually even physically there. That's not much of a problem. Now then, you have an immune response against the viral vector. So kind of like, you've caused another problem. But I think the antibody response is not as bad. The kind of cell-mediated response, where if you have this Cas13 that's being expressed inside of this cell, it's a non-native protein, and it's having different peptides being actually presented, that is a big issue that people are thinking about. And there are ways to potentially engineer around this. Different MHCs, MHCs have preferences for certain epitopes being presented, and certain epitopes on Cas13 will look more or less like your natural, actual peptides. So I think that people can use computational algorithms as well as directed evolution approaches to try to actually mutate certain regions, especially places that may or may not be more or less essential, but it's going to be potentially a challenge. I mean, all this being said, people haven't really gotten great data on whether this is even a huge problem at all. So, as we put it into more patients who have different immune responses and test these things, it'll come up if it's really an issue, and then it'll be this effort both computationally and kind of like, less rationally, just by trying things, to evolve versions that don't have as strong immune responses. Oh, there's one other aspect, which is that you could imagine just finding something like a Cas13 that is made of human parts. So I think this is Brian Dickinson at U Chicago. He had this paper called CIRTS, C-I-R-T-S, but it's like kind of like Cas13 in a sense where it binds RNA and can bring stuff with it. It doesn't cut, they can attach with it, but it's made out of human parts. That's actually the title. It's kind of a funny title. It's all these human protein domains that have been stitched together, and it doesn't work. For most things, it doesn't work as well as Cas13, but it's totally human, except for the junctions between these different domains. It's all human peptides, so that would avoid that.
Right. Oh, cool. All right, Emily.
Hi, I'm Emily. What are your keys to being so successful and impactful? And do you have any wisdom to impart for inspiring the next generation of researchers?
Yeah, I think we got the gist. Yes,
this part of it.
But just like, do we have any good advice?
Yeah, what are your keys to being so successful, and what advice do you have?
Yeah, I mean, I feel like for me, like being like in good environments, I feel like has been what's been most helpful for me. So like when we were in Fung's lab, it was just like this incredibly exciting place where like everyone was like super curious, and like there was all these like really smart people around. Like whenever you had a question, you could like go find someone. And I guess that's part of the reason why I like also being in Boston is just like this rich biomedical community. So I would say like, find what your passion is and find like a good group of people that you can work with, because like it's hard to do it alone. And I think being part of really great teams has been like the most helpful thing. And so Fung had made this great environment in grad school. I think we're trying to find that now as we grow our own group and find good postdocs and students that we can also work with, and make a really fun but like inspiring environment to work in. I think is my thought process maybe behind it.
Yeah, I think the biggest thing is the people. I mean, that's true for any walk of life. You can love what you do, and I mean, obviously we like science, but like, who you interact with every day is really makes whatever you're doing the most, either the best experience or the worst. And specifically, like, mentor Fung for us, he was just amazing. Obviously he runs this incredible environment and he's brilliant, but he also just cares about people. You can go into his office, in the middle of the day and just like talk about stuff for like 30 minutes. It's fun. So find a good mentor, I think, is the big anchor. And what that means, it's also much more about what good is, but someone who can take a personal interest in you, really accelerate your career, and they often come with a good environment. Find a good teammate. I mean, we work together really closely, and it's awesome. I mean, because we can always, not lonely in labs, there's someone to talk to, and now we have a group, and that's also nice. Also, kind of on the flip side of all that, I think when I was starting out grad school, I had a lot of fear. That an experiment would work, or that I'd do the wrong thing. I mean, stuff goes wrong all the time in science. For all of these different things that you've seen in these papers, for every good experiment, we had 9 experiments that probably failed. So, don't be afraid of failure. And when failure happens, you have to be tenacious, and don't be discouraged. So the collateral effect. This is a fun story. One of the first ways that we found the collateral effect, so we were doing this PFS screening, or actually back then we called it this CFS or something. We went through a couple iterations of acronym. So we screened it in the phage, and we did the bacterial screening, but we were also trying to do an in vitro screen, like they do with Cas9. And we were trying to incubate all these things together in a reaction with a variable region, in vitro cleavage. And we thought, okay, it'll only cut the ones with the PFS, and it'd be great, and we'll sequence it. And that reaction, it failed so many times. And we just put it together and we get no data. And we must have run like 6 sequencing reactions. First Omar was analyzing it, then I analyzed the data, and then we're like, oh, is it RNase contamination? Are we just idiots? And it didn't work, and you can obviously why, because the collateral effect, like independent of the PFS, if something was activated, it's cleaving everything. So it was actually like this horrible, horrible experiment that was like, we were pulling our hair out over. And it really turned out to be a good line of evidence that we should, let's just try this this gel where we incubate it with another thing. And it was so horribly frustrating. It was just the worst. So those moments will occur in your life all the time. And you just, it happen to everybody. So just don't be perturbed by them. And I think things will turn out good.
Yeah.
All right, we have one more question. This is probably gonna make you blush.
Yeah.
Go ahead.
Victoria, yeah, so as young scientists, we really look up to you guys and are completely fascinated by, how much you've completed and how old you are. And so my question is basically, how does it feel to be listed on Forbes 30 under 30?
I think it was okay. I actually didn't like the photo that much. I mean, but it's nice. I mean, it's a good community, and I think we've met a lot of interesting other like scientists and people in healthcare through it. So that's been kind of nice. But functionally, it really has no effect on our day-to-day or like pushing forward the science. I think it makes it helpful. I think we've seen as we're independent to maybe raise more funding. A little bit, but in general, getting funding, I think, for your work is really hard.
Yeah.
But yeah.
I don't know, it was cool, but I think it's not as, you feel good when you get it, but like these things, like any award, or also bad things that happen to you, it kind of like evens out over time. And like, more importantly, it's like, surround yourself with good people that make you feel happy. And like, there's plenty of things that we haven't gotten. There was actually just a big award that we both didn't get, and that would have been nice because we did some funny for laugh. But these things, you just kind of like have to see it as like, there's a lot of things in life, and I don't think chasing achievements eventually does make you happy. I don't know. It's more about like the long term and like finding what really satisfies you. But yeah, it was neat for a bit.
Yeah.
Well, that is all the questions that we have. I just wanted to thank you for your time, but I also wanted to let you know that the work that you're doing isn't just for the scientific community when we think about our grant reviewers and the people reviewing our papers, but I want to let you know that you're really inspiring the next generation of scientists, and we really appreciate having the opportunity to actually interact with you. This has been fantastic.
Cool, thanks.
Yeah, this was
so much.
Super awesome. We have one question at least, which is, I have to remember the key command. We wanted to take a screenshot of the actual thing. Do you know exactly what it is? Is it Shift Command 3? But like, I think it's perfect. So yeah, I don't know. You guys can like wave. What was your idea for this? Like,
I don't know if you guys wave. We can just take a
wave.
Yeah.
Cool. You got it. Yeah, there we go.
Oh, did it work?
Yeah. So, but no, thanks for the opportunity. Thanks for reaching out. This is like super fun for us. So, and you guys are inspiring.
Well,
we like the next one.
We thought there was a chance you might actually do this because you're not super famous yet. So maybe in a couple of years you wouldn't think twice, but we really appreciate it. So thanks a lot, and good luck with your continuing research.
Thanks. Yeah.
Thank you so
Have a good day.
much.
All right,
Thank
bye.
you.